Welcome to the discussion session for week 5. So, today, as we normally do, we will first be going over the topic that was covered, the topics that were covered in the previous week. And we will first look at what were the assignment questions that were as part of the week 4 assignment, and most of the answers were the same. And following that, we'll also be looking at the topics that were part of week 5 and go over some sample questions for the same, which is the pattern we follow normally as well. So, first of all, for the assignment of week 4, we were asked that in question 1, we were asked that based on endosymbiont theory, which organelles in the cells of a zebra have prokaryotic origin? Under that, the options were peroxisomes, mitochondria, chloroplasts, or both B and C. So, if you remember the way we had discussed endosymbiont theory, according to that, um, an initial primitive cell had taken up prokaryotic cells, which particularly included the um, uh, archaebacteria and photosynthetic bacteria. It is believed that over the course of time, they have evolved to become what we now understand as the mitochondria and the chloroplast in the advanced, not advanced, but particularly complex eukaryotic cells that we are familiar with today. So this was a theory that lacks a lot of supporting evidence. There are certain arguments that support it and certain arguments that go against it. But the endosymbiont theory is one of the ideas that is currently explored. And while it is up for debate, it is something that does explain the evolution from the prokaryotic world towards the eukaryotic world. So according to that, the organelles which are believed to be of prokaryotic origin are mitochondria and chloroplast. But when we are talking about cells of a zebra, which is obviously not a photosynthetic organism, we only talk about mitochondria. Therefore, option B was the correct option. So you need to make sure that if the question is mentioning a certain type of organism, then you are narrowing down your options accordingly. Then in question 2, we were asked that which of the following is a common characteristic of both prokaryotic and eukaryotic cells. So we had discussed some of the common differences between the two, and among them size, DNA organization, use of ATP as the form of energy or the energy currency of the cell, and the use of mitochondria are being mentioned as possible options. So when we talk about prokaryotes versus eukaryotes, there's an obvious difference in size. DNA organization is much more complicated and much more organized and compact when we talk about eukaryotes. However, prokaryotes are believed to sort of contain an unorganized nucleoid-like structure, and therefore option B is also not correct. ATP as the form of energy is the common characteristic between the two, while there are other sources of, not sources, but other ways of transporting electrons or basically carrying energy around inside the cell, even in case of eukaryotes, like there is GTP and there are other molecules as well. ATP is the main energy currency within the cell, and that becomes a common characteristic between the two as well. Then the next question asked is that the resolving power of a microscope is limited by refraction, retraction, reflection, none of the above. So the answer to that would be refraction, which is why you don't get if the resolving power of the microscope is not very high, you will not get crisp images. Instead, you end up getting very diffused or unclear images under those circumstances. Then the next question was about fluorescence. We had discussed this. I guess this was even one of the sample questions that was there. So fluorescence is the ability of a molecule to do which of the following. So option A was about absorbing light of one wavelength, emit light of shorter wavelength. Of course, this is not correct. If you remember the diagram, it's often known as the Jablonski diagram, where different energy levels are being shown and how upon being excited by some sort of radiation, some sort of an energy source, electrons will go from one energy state to from the lower energy state to the higher energy state, and based on how they come back to the lower energy states, different types of emissions can be observed. Like there is fluorescence, which is what we have talked about. There is phosphorescence as well. So that diagram would be a good reference for your understanding. So the concept behind it remains the same, that the molecule will absorb light of a certain wavelength and it will emit light of the same or of the longer wavelength. So option B is correct. Then in question 5, we are asked that which of the following cell types contain plasma membrane? E. coli cells, lettuce cells, viruses, or both A and B. So if you remember the brief introduction that we had into the different components of the cell, I had shown that that any living organism needs to have this boundary or needs to have this peripheral what should I say, like a sort of covering on the outside, which is the plasma membrane. And since viruses are sort of debated as whether they are living or non-living, in fact, a lot of arguments consider viruses to be on the verge of both because they are capable of replicating when they are inside their host, but they are not capable of just surviving by themselves. So they are dependent, and therefore they are said to live on the verge of living as well as non-living. However, if you've had some basic introduction into how viruses actually are, the structure of viruses, then you'd find out that they do not actually have a plasma membrane. They often contain, many a times, they consist of a lipid sort of coat or a covering on the outside, apart from their protein structure, the normal structure that most viruses have. A few of them also have the outer coating, but that's not necessary feature for all of them. And they certainly do not need a plasma membrane. So viruses are not the correct option. But other than that, since all living organisms should have a plasma membrane, both E. coli and lettuce cells would have the same, and option B would be correct. Then in question number 6, we're being asked that the first compound microscope was made in the 16th century by Robert Hooke. Galileo Galilei, and Leeuwenhoek or Zacharias Janssen. Then we discussed this that Zacharias Janssen, who was believed to be, who belonged to some family of spectacle makers, he is considered to be the father of microscopes, and he made one of the most primitive microscopes very early on. I think sometime in 1700s or 1800s, the first microscope was made, which could only zoom up to, like in today's world, it seems like a very little magnification, but back in those days, it was an excellent venture. And there was Zacharias Janssen was credited with the discovery of the initial microscope. And it was, oh, sorry, it's the question only in the 16th century he had made this discovery. Um, then in question 7, we were asked that a major component of lysosomes is A, calcium, B, glycosylation enzymes, C, hydrolytic enzymes, or D, water. So if you have gone through the questions that the pointers that were there about each of the different organelles, then you would find out that lysosomes, they are considered to be the digestive bags within the cell. They are capable of chewing up or breaking down whatever is inside the cell if it is required to do so. And therefore, whenever you talk about breaking something down, what is required, you need a hydrolytic enzyme. As you can understand by the name, it is lysing something, which means it is breaking something down. So that is one of the major functions of the lysosome. So it needs to have hydrolytic enzymes. So on the other hand, if you look at the other options, calcium as well as glycosylation enzymes are usually associated with the endoplasmic reticulum. It is sort of under the purview of vesicular trafficking, and water is while inside the cell, water is obviously an available molecule, but it is not the major component of lysosome. So to say, there an acidic pH needs to be maintained, and that is necessary for these hydrolytic enzymes to be able to work. So yeah, that was question 7. Then in question 8, we were asked that the nuclear pore complex selectively allows the export of molecules to the ER, export of molecules directly to the cytoplasm, both import and export of molecules to the cytoplasm, and import of molecules from ER. So as you can understand by the name, the nuclear pore complex is a component of the nucleus and the various pores or the many pores that are present on the nuclear membrane. So the purpose of it is, apart from other things, one of the major purposes is to allow the translocation of the newly synthesized mature mRNA from the nucleoplasm or from the nucleus into the cytoplasm. And apart from that, it has to allow whatever is required for any, say, so any particular protein that is a nuclear protein after it has been synthesized in the cytoplasm by the ribosomes that are present in the cytoplasm. Such a protein, it needs to be brought back to the nucleus, right? So there is some sort of, you can think of it as an address. Proteins, after they are synthesized, they carry an address or a sort of a tag which is necessary for them to be directed to their current location. So in case of nuclear proteins, such a signal is known as the NLS, which is nuclear localization signal. What it effectively does is, after a nuclear protein has been synthesized, that protein is brought back to the nucleus. So the nuclear pore complex, because that is the only sort of entry-exit point for anything to come into or exit from the nucleus, they are involved in both import and export of molecules from the nucleus to the cytoplasm or vice versa. So option C is correct. Then in option D, we're being asked that which of the following features supports the idea of a chloroplast being an endosymbiont. So we discussed endosymbiotic theory in the first question as well. So the chloroplast is a double membrane bound organelle. Yes, that's of course the idea of endosymbiosis. It carries its own DNA, that as well. Ribosomes in the chloroplast are similar to those in prokaryotes. Yes. So all these three features together amount to the fact that they are all supporting facts, or you can say supporting arguments that are usually given in favor of endosymbiotic theory. So option D is correct. Then in question 10, we were asked that which of the following statements about vacuoles is true. I don't think we discussed vacuoles in a lot of detail, but you'll be able to get some idea. The first question is that vacuoles provide turgidity and rigidity to animal cells. So while the initial part of the option might seem correct to you, as you can obviously understand, animal cells do not have vacuoles under normal circumstances. So the purpose of turgidity and rigidity is um, much more important when you talk about plants, because that is necessary for maintenance of the shape of plants. One of the major ways in which the turgor pressure and the shape of the plant cell is maintained. So it is relevant to plants, but certainly not for animals. So option A is not correct. Then the fact that they regulate cell division and help in plant growth, also not true. They help maintain osmotic pressure in the cells is certainly correct. And vacuoles only appear when plants are in stress. No, that's also not something that's true. So option C was correct. So these were the questions from week 4 assignment. In case anybody has any doubts, please bring them up. I'll try to clarify them. And before we get to the week 5 questions, we will first go over the topics that were part of week 5. So if you remember, if you've gone through the lectures, then week 5 was going into a little more depth about cell biology topics that we have already briefly touched upon in the previous week lectures. So here the first idea is to understand the compartmentalization that exists within the cell, and under that the broad topic obviously becomes the boundary of the cell, which is the boundary of the cell and the inner compartment creator. So what is creating that compartment is obviously the lipid bilayer or the cell membrane that is present. So when we talk about the cell membrane or the cell wall, either of the two, they are both very relevant structures to maintain a difference between the cell interior and the cell exterior. So here, what is the initial definition? If you look at it, says that the cell envelope may be defined as the cell membrane and a cell wall along with an outer membrane if one is present. Now, the concept of outer membrane is majorly restricted to prokaryotes, particularly certain bacteria, even not all bacteria, which you've obviously gone through. And the envelope or the outer covering consists of mainly the cell wall and the cell membrane or the plasma membrane. The cell envelope or cell wall is seen in bacteria as well as plant cells, and the cell wall is like I think I said this in the previous interaction session as well. The difference between a cell wall and a cell membrane is the fact that how permeable or how rigid their structures are. So if a structure is um, not allowing anything to enter, or is it's in the name itself, right? If something is a wall, it's very rigid. It's when we say rigid, it means in the sense that it is physically tough and it is also responsible for restricting entry and exit of molecules. In comparison, if you talk about the cell membrane, it is a lot more permeable, lot more um, what should I say, a lot more accessible to a lot of other molecules. So here the cell wall is a tough, flexible molecule, but sometimes fairly rigid layer that surrounds cells. And the location of the cell wall is obviously exterior to the cell membrane, and therefore its major structure, major function goes towards structural support and protection of the cells that it is surrounding. When we talk about the bacterial cell wall, you can see here a cross section of a bacterial cell has been shown. So you see the outer covering, which is a capsule. So a capsule is again not something that is present in all bacterial cells. The capsule is, you can think of it as an additional layer of security that is present or in some species of bacteria, because that gives them added protection, that gives them more strength, more chances of survival, and um, it is an added advantage in a lot of different types of environments, which allows those bacteria to persist despite whatever environmental conditions might surround that place at different times of the year. So it's an added layer that is present. Apart from that, just beneath it, we see the cell wall. Then comes the plasma membrane, and then along the plasma membrane, we see a large number of villi, which, as was discussed in the lecture as well, is majorly concerned with cell-cell contacts, sometimes in adhesion, sometimes in um, taking up a lot of maybe some food material or some contact with the environment. And you can think of it as equivalent to villi in the intestine, in the human intestine that you might have already come across, that villi increases surface area of absorption of nutrients. And in comparison, villi, they are not directly related to each other, just for our understanding that the purpose of the two in their own host systems is kind of similar. Villi or pillars are more centered towards increasing the surface area, which is in contact with the surrounding environment. So that is the job of the pillars. And inside the bacteria, you see the cytoplasm, which has a lot of interspersed ribosomes necessary for synthesis of proteins that we just talked about. And you can see the genetic material, which is sort of in a haphazard, unorganized state, which is obviously why it is not as compact or it is not as well defined as in case of eukaryotes, and is usually only considered to be a nucleoid. So that is what we see here. And apart from that, the flagellum, which you can see here, is an extended sort of structure which is present in bacterial cells, and it is concerned with providing the bacterial cell a lot of added motility, and it allows the bacterial cell to propel itself forward and move through whatever material it is present in, whatever medium, maybe it's a viscous medium that the cells are present in. So it allows them to propel through that. Um, then come bacterial cell walls. So because we talked about the fact that not all bacterial cells um, uh, have a outer membrane, or they have difference in their cell walls. Let's think of it that way. So they are majorly based on the presence of that outer membrane or the absence of the same. They are classified into gram-positive and gram-negative bacteria. So explains this a little, slightly in a little more detail. We'll look at the idea of gram staining, but before that, let's just go over the ultrastructures. So most bacterial cell envelopes, as has been shown here, are in two categories. When you look at the gram-positive bacterial cell wall structure, it has a plasma membrane, which, as we have said, is present in all living organisms. And just on top of it, there is a layer of there's a layer which is called the peptidoglycan. So without going into much detail, if you just look at the name, peptidoglycan, it means there are peptides and there are glycans. So there are some proteins or amino acid like structures, some peptide bonds or something like that, and there are glycans, which means there are some sugars. So basically, there are a lot of sugars which are arranged in a cross-linked manner. These are, if you're interested, read up on it. There are NAG and NAM residues which are present and they are cross-linked to each other. And there is an incredible mesh-like network which is formed, which gives like a thick layer of peptidoglycan on the surface of these cells, or on the surface of gram-positive cells. In contrast, while the same materials make up the peptidoglycan in the gram-negative cells as well, the peptidoglycan layer is much thinner. And instead, they have a very thick outer membrane, which is made up of some other components. It has teichoic acids. It has some, I think, it's called the LPS layer, so lipopolysaccharides, etc. It has a different composition. I'll have to go over it once more. But the outer membrane is a separate structure which is present and is a lot less permeable than the peptidoglycan, which makes them even more dangerous pathogens when they infect us. So while pathogens can be gram-positive and gram-negative as well, these are more resistant or more, you can think of them as better survivors in the host environment in a lot of ways. So this difference in structure is understood based on the gram staining characteristics. So before we get to that gram staining part, let us understand this. The peptidoglycan, like we have just discussed, is a single bag-shaped, highly cross-linked macromolecule which surrounds the bacterial cell membrane and is responsible for providing rigidity as well as, to a large extent, protection to the bacterial cell. It is huge. So it is said to be billions in molecular weight compared to thousands, which are compared to proteins, which are thousands in their molecular weight. They consist of glycans, which are polysaccharide backbones that have NAM and NAS, which I was just talking about, that are N-acetylmuramic acid and acetylglucosamine, and they have peptide side chains, which have D and L amino acids. And sometimes in certain first regions, that I think the residue in that crosslink is diaminopyruvic acid. So it's a very distinctive structure. There's not much to explain there. You can go over it if it is something that is interesting to you. And these side chains become crosslinks. So here you'll be able to see that structure. These residues that you're seeing, if we zoom into the structure, you'll see this M and G. So that is NAG and NAM, or N-acetylglucosamine and acetylmuramic acid. And these colored extensions that you see, they are the peptide side chains that we're talking about. And you can see that there is a cross-link between the fourth and the third residue between the two. So that gives a very intense, intensely cross-linked structure that is present here. So this you see is an E. coli peptidoglycan that has been shown here. Here you see a Staphylococcus aureus peptidoglycan, and there is a pentaglycine bridge, which is linking these. So these are very, it's a lot of detail into the structure of it. You don't necessarily need to remember that much detail. You can keep in mind the residues that make it up and purpose of the peptidoglycan. What is the difference between gram-positive and gram-negative? So when you look at the gram-positive cell wall, as we were just talking about it, here you see, so again, think of this as a cross section, that if you had a bacterial cell and you sort of chopped it in half, what you'll be able to see. So here you see this is a very detailed and very well represented schematic of the cytoplasmic membrane or plasma membrane, as has been shown here. And if you look very carefully, you'll see it's a lipid bilayer. There are two layers of lipids. So you can think of them as two mats that are right one on top of the other. And you can see some transport proteins which are spanning the two that have the job of allowing transport of whatever specific molecule it is capable of binding to from the outside to the interior of the bacterial cell. And above it, we see that extensively cross-linked structure that we were just talking about, the peptidoglycan, and it is made up of these particular amino acids. So this is also, I think this is a electron microscopy image, most probably a TEM image, I think, I'm not sure. So the difference between SEM and TEM, I think we came up with it on the microscopy class as well, that STEM or Scanning electron microscopy allows you to see the surface details of whatever molecules you're trying to study. Versus STEM, transmission electron microscope, allows you to see the ultrastructure, that is what is inside. You can study the cross section and get into the nitty-gritty details of it as well. So here you can see the individual layers, and this is a representative image of what a gram-positive cell wall looks like. In comparison, if you look at the cross-section of the gram-negative cell wall, the initial part is the same. This is the plasma membrane, or the inner membrane or lipid bilayer. After that, there's a little bit of space, which is commonly known as the periplasmic space. Periplasm is, if you take it quite literally, it is said to be adjacent to the plasma. So right next to the plasma membrane, the region or the um, it is not a vacant hollow sort of space. There is periplasmic fluid in there as well, but it is not really it's not really performing very definite functions. You will find a periplasmic space for mitochondria as well, which is again another idea that could support the concept of endosymbiotic theory, that there is a little bit of space between the mitochondrial membranes, outer membrane and inner membrane. So that's a separate topic, but yeah, the periplasmic space, and on top of that, we see the outer membrane, which is a lipid bilayer again, and the outermost region of it has a very intensely decorated. Like you can see how extensively decorated, for lack of a better word, decorated the outer membrane is, and it has a lot of like proteins which are covering it. So this LPS layer is one of the major reasons why gram-positive, gram-negative bacteria, like we just talked about them, are much more dangerous, much more severe pathogens. So whenever you study this, keep an eye on a few examples for the same for gram-positive and gram-negative bacteria. And again, this is the electron microscopy image for the same as well. And you can see that there's a little bit of a capsule, which we were talking about there. And the rest of the structure, the peptidoglycan cell membrane is the same, just the outer membrane is the point of difference between the two. Now comes the idea of gram-positive and gram-negative bacteria, that how would you understand it? Because if you just go for microscopy, they all look like bacterial cells. They might have different shapes. They can be a bacilli, they can be pockets, they can be vibrio, flagellated, etc. Those differences would be there. But the nature of the two bacteria are difficult to understand. So for that, while the PPT itself also has the same details, I have just included another slide because it explains it really well. So in case any one of you has done gram staining, you'd already be familiar with it. So gram staining was given by, I think, the scientist credited with the same as Hans Gram, and came up with this idea that you could use two different types of stains. Which means there is one mean or primary stain, and there is another counter stain, which is used for the purpose of distinction. So the primary stain in this case is crystal violet, and all bacteria, when you use crystal violet, irrespective of their nature, will initially be stained in that bluish purple sort of color, which is what you see here, that during this step, if you just observe these cells under a microscope, they would all appear blue in color. Once this is done, you stain it for 1 minute and then you wash it with water. And once this has been done, you use a mordant. So mordants are chemical entities basically that allow for better binding between the coloring agent and whatever it is supposed to bind to. So in this case, it is grams iodine which is used, and what it does is it enhances the binding of crystal violet and it forms a crystal violet iodine complex, or a CVI complex. And what happens is that while you are visibly not able to see the difference between the two, within the cell, the binding between gram-positive in gram-positive bacteria, the binding between the two is enhanced. Following that, you wash off that iodine for 1 minute, and then you wash off that iodine, and then you add a decolorizer. Now, decolorizer is usually acetone or ethanol. So what it does is, as the name indicates, it's a decolorizer. So it gets rid of any non-specific color binding or any loosely bound stain. And yeah. So it gets rid of the loosely bound stain and it washes away the primary stain from the gram-negative bacteria. And following that, counter staining is performed with safranin or Carvol fusion. And this counter stain, what it does is the bacterial cells that are no longer colored, which is what happened during the previous step. Okay, I've got to mention it. So during the decolorization step, only gram-negative bacteria become colorless. That is because the color has not been, you can get into the detail of it, that where exactly does crystal violet go and bind. I'm not getting into it right now. You can look it up for your own interest. But in case of the gram-negative bacteria, because the binding is not as intense, the color is gotten rid of, and therefore gram-negative bacteria become colorless. Following which, when a counter stain is used, which in most cases is safranin, or as per gram stain protocol, it is safranin. It stains these gram-negative bacteria red in color. And after the staining has been performed, again, washing is done. Then you mount it on a slide and observe it under the microscope. And when you observe it under the microscope, you can actually see the difference that is between the gram-positive and gram-negative bacteria. That is a representative image is actually shown here. So here you can see that these sort of round sort of bacteria, the coccus shaped bacteria, there's a difference in the morphology as well. But the ones that are more rod shaped, the bacilli shaped ones, they are appearing to be gram negative, whereas the coccus shaped ones or the roundish ones are gram positive in their nature. So this is one of the preliminary microbiology techniques that are often taught in preliminary courses, and they are till date they are used in a lot of pathological conditions as well. While it is not a very um, like a very hugely used or extensively used pathological technique, it is still very useful, and it could be particularly useful if you're trying to understand some new bacterial strain, something that has not been studied before, or that somebody has discovered on their own for the first time. So one of the first things that is done to understand how that bacterial cell actually looks is gram staining. And then, of course, you go into other more advanced microscopies techniques to see what it looks like, what are its features, what are its expression, what is it pathogenic or non-pathogenic, and so on and so forth. There are many good tests for that as well. So we talked about the peptidoglycan layer, which is the layer that is majorly involved in not majorly involved, which is the layer that protects bacterial cells. Let's say directly. However, when we talk about plant cells, their cell wall composition is entirely different. In fact, even in bacteria, if you are interested, sometimes go and look up what archibacterial cell walls are like. They are actually quite different. They are not exactly the same, and they had much more rigid cell walls, which, if you think about it just from a speculative point of view, you'll understand that considering the conditions that archibacteria are believed to have survived under, which were much more extremes of temperature, of pressure, of extremely hot, extremely cold in very volcanic areas, etc. So those bacterial cells needed to have uh, much tougher defenses in place in order to be able to survive in such locations. So their architecture or their cell wall architecture is a bit different compared to that of the modern day or the more commonly available bacteria that or the eubacteria that we talk about. Compared to them, there's a lot of difference between the two. Coming back to plant cells, on the other hand, first of all, the composition is very different. While we talked about peptidoglycan made up of NAG/NAM residues and peptide side chains, in contrast, plant cell walls are made up of cellulose, polysaccharides, and proteins. Cellulose, hemicellulose, polysaccharides, and proteins. So in new cells, initially the cell wall is quite thin, not very rigid, which is something that is, you can think of it as necessary because after a cell has just been synthesized, it needs to grow up. So like if a baby is born and you immediately, you know, tape it and try to restrict it, of course, the baby would face a lot of hindrance in growing up. So just a very loose example, but yeah, similar to that, because expansion in size is one of the initial features that is required for plant cells in particular. And because, as you understand, that vacuoles would also have to increase in size. We talked a lot about vacuoles in a little while ago, and they are responsible for exerting the pressure that maintains rigidity and turgidity of plant cells. So to accommodate all of those changes inside the cell wall needs to be something that should undergo some amount of dynamism over the growth of a plant cell, over the time of growth of a plant cell. So when a cell, however, is fully grown, it might retain its primary wall, sometimes might end up thickening the same, or there's another, like these are just two possibilities. Either of the two can happen. It might just deposit new layers of different material, which would end up becoming the secondary cell wall. Uh, on the whole, what happens is that each cell's cell wall interacts with its neighbors and it forms a tightly bound plant structure. And despite the rigidity of the cell wall, chemical signals as well as cellular excretions are allowed to pass between the two. So this you see is an example of a parenchymatous cell. And it provides the cell wall here, which provides the rigidity to the structure, has been shown in green. So if we talk about the cell wall structure, it has been shown here. So like the ECM, the plant cell wall connects the cells to its tissue, connects cells into forming tissues, and it signals a plant cell to divide and controls the shape of those organs as well. So it has the arrangement is such that it has cellulose microfibers, which are okay. Also go and look up what cellulose is made up of. What are the two types of um, entities or sugars which make up cellulose, and this material is embedded in a matrix of pectin and hemicellulose, forming a cell wall that is approximately 0.2 microns thick and completely coats the outside of the plant cell. So here you see a representative image of the same as well, where you have the plasma membrane, and on top of that you have the primary wall, and it can happen under certain circumstances when a plant cell has grown for say 50 years or so. When it's an older plant, then more deposition can occur and it can lead to the formation of a secondary cell wall. It can vary from species to species. So yeah, this is what a representative image of how fibrils, microfibrils, and cellulose are arranged in cell walls. So just a representative image of the cellulose fibril, and from it the microfibril, and what it is, the monomer or the smallest unit of it is cellulose at the end of the day. So this is another representation of how complex and how intricately arranged cellulose microfibrils are in the primary cell wall. And this, I think, is an example of a carrot cell, and just its ultrastructure has been studied there. So cellulose effectively forms the bulk of the plant cell wall, and the microfibrils themselves become embedded in a matrix or a sheath. So cellulose embedded in a sort of um, mesh or a matrix, which is made up of hemicellulose, pectins, glycoproteins, various enzymes, lignins, cutins, suberins, waxes, which are present in the cells on the outside of the plant body. So you can see that even within plant cells, there is a variation dependent on where they are located in the within the plant body. So if they're located towards the outside, most of the times plants have an extra waxy coating, which is made up of cutins, suberins, and waxes, as has been indicated here. And in those cases, the plant cell wall is surrounded by an additional layer. So we'll come to this. We'll talk about the secondary cell wall. So under normal circumstances, when they are very actively dividing cells, or when some cells are very metabolically active, example here as is given for photosynthetic cells or cells that are secreting something constantly, which is necessary for the plant to function normally, they usually would have only one layer, which would be the primary cell wall. In contrast, when we talk about cells that are more involved in providing some strength, which are associated with compressional strength, such as when we talk about wood, the components of wood, they keep on adding additional layers inside the primary wall to form the secondary wall. I think there was another concept, which is there was a rosewood and some other, sorry, I cannot recall it at the moment, but depending on the season, the type of deposition that occurs in the secondary cell wall can vary, and that ends up affecting the type of, the quality of wood that you would get from such a tree. And depending on that, of course, because wood is one of the major sources that is used for construction of furniture, not so much today, but yeah, it does determine how strong such a such an object or such a furniture product would actually end up being. So these are all applications of science in real world in understanding how strong that plant would be and how expensive such a product would have to be priced. So here I think there was a video on cell walls, which if you're interested, you should certainly go ahead and look it up. I think it was in the lecture as well. So that discussion was majorly restricted towards cell walls. But when we talk about the cell envelope, it does not just include the cell wall, but the next and in fact the major contributor towards the building this boundary around cells is of course the plasma membrane. So here you can just see a representation of what um, of a sort of scale across not necessarily organisms, but different types of studies that have been conducted. So you go from atomic scale from 100 picometers to eukaryotic cells, which are around 100 micrometers in size. Some of them can be under certain circumstances. Then you have the organelles, bacteria, viruses, proteins, lipids, and small molecules, and the type of microscope that you would actually need to visualize the same, that would also vary. So electron microscope or light microscope or something that is just visible to the naked eye, that will all be dependent upon what sort of object you are trying to actually visualize. So here you see, okay, so when we talk about the plasma membrane, uh, it used to be one of the biggest mysteries of the biological world at one point of time, and there are many different types of ideas that came into it. We are not getting into detail of it. We'll come to the one that is the most accepted and the currently most well-studied idea of the plasma membrane as well. Initially, it was believed that it sort of is like a sandwich. It was first thought to be a completely rigid structure. Then, as time evolved and as people got more and more advanced with their technological prowess, more and more was understood about the cell membrane. So you can see that the previous idea, and this is sometime in 1974, it was just believed to be like the presence of proteins within the lipid bilayer was considered to be much like carrots or other rooted vegetables that are present within the soil. So it was as simplistic as the idea could be. Today, we know in extensive detail what is making up the membrane, how they are arranged within the membrane. Whether they can only stay localized at one region, whether they can move around, whether they can switch from one side to the other, and a lot more detail. So here it says that the commonplace view now is that the average bilayer lipid viscosity is similar to that of olive oil. A more exotic standard is the viscosity of crocodile fat on a warm summer's day. So this is not really relevant, but the idea is that when we talk about viscosity, it means that the bilayer, while it was initially believed to be this strong, rigid layer that is protecting the cell from the outside and is maintaining the idea of what the cell is from its exterior, that view has changed, and now we understand that it's actually not that rigid. It's a soft, viscous, fluid sort of a layer. So currently what we understand is it looks like this, that if you take a cell, a eukaryotic cell, or any cell for that matter, in this, however, is a representation of the eukaryotic cell, and you just chop it in half and you take a section of the membrane and you look at it at this detail, at this level of detail, you will understand what exactly makes up the plasma membrane. So first and foremost, if somebody asks you what is making up a plasma membrane, then the direct answer is coming to the presence of phospholipids. They are the major components there. Phospholipids, we talked about it, I think, a little bit in detail. They are, as the name indicates, they have a lipid moiety and they have a phosphate moiety. So if you just look at the simple representation here, you are seeing these little red colored head moieties, the hydrophilic heads and the hydrophobic tails, which are localized towards the interior. And that maintains the nature, the outside and the inside of the membrane is maintained because of the nature of the chemical entities which form the phospholipids, which are making up the plasma membrane. So because these are because the membrane is not made up of just one huge sheet made up of a simple material, but is instead made up of tiny, tiny little molecules that all together form this wobbly sort of sheet. Therefore, there's a lot of fluidity or a lot of movement that occurs within the plasma membrane. Now, why the idea of fluid mosaic model? So the fluidity part of the membrane was well understood that the membrane is fluid in nature. You can see proteins that are localized within the membrane to be moving about because of how these proteins are actually interspersed. If you've ever, I'm sure most of you would have come across mosaic flooring, right? Which used to be more popular a few years back, 10, 20 years back. That was what you would find in everybody's houses. So that had a sort of whitish background within which little colored chips or stone chips used to be usually present, and it gave a like a really beautiful pattern. Similar to that or analogous to that, it is believed that it is a phospholipid background within which proteins are interspersed in different or embedded in different regions, which gives a mosaic-like appearance if you look at the cell from a bird-eye view. So that gave the concept of fluid mosaic theory, of fluid mosaic model. If you're interested, check out who exactly gave this idea and whether they have received credits and laurels for it or not. So here you see that there are a number of different types of proteins as well. You're seeing some integral proteins, like which are mostly globular proteins. You are seeing some surface proteins. You are seeing some alpha helical integral proteins. So there are, like you saw here, this is also an integral protein. This also is an integral protein. However, there's a difference between how they are present. While the globular protein seems to be just directly inserted, it is forming a globule and is inserted directly. The alpha helical protein is quite different. It forms a spiral or helical structure, and it has some extensions both in the extracellular space as well as in the cytoplasm. So this, how is this happening, is a question you might ask. So uh, it is a little bit beyond the idea of this course, but if you remember, I had talked a little bit, very little bit about some co-translational folding in one of the earlier classes. So certain proteins, after they have undergone certain mRNA, after they have been transcribed, right after transcription, there is a little bit of folding which can be initiated for certain proteins. So what happens is as soon as the mRNA is getting translated and the protein or the polypeptide chain is getting synthesized, it immediately takes up whatever shape it is supposed to do. Why this happens and what all are involved in it is a whole complicated topic that would be a much more advanced concept, but it is something that can happen, and the ER is one of the major sites that is very, very crucial to this process. So while they are both integral proteins, their nature, their folding, their appearance, everything is quite different. Other than just proteins and phospholipids, we can also see a lot of other molecules as well. So you can see certain, okay, apart from these two integral proteins, you can also see some protein channels, which, as the name indicates, they are sort of, you can think of them as sort of pipes, which through which you can pass whatever ions or whatever molecules you want to, like glucose, etc. So protein channels act as transport proteins. You have other glycoproteins as well. You have peripheral proteins. Peripheral proteins, if you look at it, their localization is restricted to one leaflet. Leaflet basically means that if you think of this as the lipid bilayer, there are two layers. The one on the one of them, which is facing towards the outer side, outside the cell or extracellular fluid, is said to be the outer leaflet, while the other one, which is facing the cell interior, is called the inner leaflet. Sorry, but when a particular protein is restricted to only the inner leaflet, then under such circumstances, it is called as the peripheral protein. Compare that to the surface protein, which does not have any embedding between the between the phospholipids, but it is just present on the membrane surface, so that makes it a surface protein. We also see that right next to the just immediately beneath the plasma membrane, there is this sort of noodle-like structure that you can see very faintly. That is an example of filaments of the cytoskeleton. So we'll be coming to the cytoskeleton in a little bit, in a not extensive detail, but yeah, some idea about it you will get. So they are present right adjacent to the membrane, and they have a lot of different functions, very diverse ones. But among them, one of the major jobs that they do is that they maintain the shape of the membrane and effectively the shape of the cell as well. So they are present in immediate close proximity with the plasma membrane, as has been shown in the figure as well. Now comes the idea that membrane lipids are actually amphipathic molecules. So whenever you look at membrane lipids, they basically have a hydrophilic tail, hydrophilic head, and a hydrophobic tail. So what it means is effectively that the heads which are formed, they are forming a sort of not exactly globular, but a sort of concise, compact structure, and they're facing water or they're facing the aqueous solvent. In contrast, the hydrophobic tails are towards the interior and they're maintaining that lipid bilayer, like we talked about it. And the hydrophilic heads, which are polar in nature, are also forming hydrogen bonds with water, while the tails are getting excluded by the water molecules and they are maintaining the cell membrane. And this is a little more detail into how the membrane lipids are made and what are the major classifications of the different subclasses of the membrane lipids. So we have phospholipids, which is here shown as phosphatidylserine. So you have the two hydrocarbon tails, which is getting attached to the hydrophilic head, and to that you have the phosphate moiety, which is attached. And on top of that, the blue colored box that you're seeing, that is for serine. If you remember the amino acid, you don't necessarily need to remember it all, but yes, the above molecule is serine. So this entire moiety is forming phosphatidylserine, which is one type of phospholipid. Other than that, you can see cholesterol, which is a sterol. And if you remember, there was a little bit of discussion in the lecture about lipid rafts. So raft, if you know about it, is sort of like an open boat sort of structure, like a floating support, which is often used by people when they're going out into some sort of water body just to float on the surface. So somewhat similar to that, lipid rafts are sort of these support structures that are present in random locations within the cell, which are necessary to hold certain proteins in certain areas. And they are majorly, one of the major components for the same is cholesterol, which is shown here. And apart from phospholipids, the other types of molecules that are also very major contributors to the plasma membrane are glycolipids. So glycolipids are one example here that has been shown here are galactose cerebroside. So you can see that it has galactose sugar attached to the hydrophilic head instead of phosphatidylserine as was attached in the previous case. So cells can actually contain hundreds of different lipid species that can be categorized into three main classes: glycerophospholipids, sphingolipids, and sterols. Further additional complexity of eukaryotic lipids is generated by many possible modifications of the hydrophilic head group. So the head group can keep on getting more and more complicated, or the hydrocarbon chain can also vary. You can have saturation or unsaturation depending on that. What would vary is how fluid or how rigid that particular membrane is. If the percentage of cholesterol is different, the membrane fluidity would vary, and so on and so forth. So here you see glycerophospholipid versus sphingolipids. Sphingolipids have a different background. They have a sphingosine background. By background, I mean the carbon chain that you see here. You can see that there is a little bit of difference in the representation whenever there is some sort of saturation which is coming about. Saturation means a double or a triple bond which is coming about. There is a change in the structure. It restricts the movement of such a molecule and it makes it more rigid, and it introduces a kink in the phospholipid basically. So among all of the phospholipids that have been identified, phosphatidylcholine is assumed to be the most abundant phospholipid in cell, not assumed, but it has been found to be the most abundant phospholipid in cell membranes. So here you see that what I was talking about actually, that if there is sort of a kink, then the hydrocarbon chain is it whenever there is saturation, unsaturation, which means that there's a double or a triple bond, what it does is, while normally the two hydrocarbon tails are represented as two straight lines, whenever such a bond is introduced, there is a tilt because it is now restricting the possible conformations or the possible orientations in which that particular hydrocarbon tail can exist. So the representation in both cases is quite different. Uh, yeah, this you see is a just schematic representations of phospholipids, sphingolipids, cholesterols, and polylipids. So what happens in the bilayer is okay, we've discussed the fluid mosaic model, but basically what they are talking about is how phospholipid bilayers are formed. So, or why at all they are formed. So the idea behind it is that when you have, if you just take a large number of phospholipids and you just add it into water, what it will always try to do by the laws of thermodynamics is that it will arrange itself into a tiny little ball. Uh, and it will seal itself from the external environment. It will try to exclude as much water as possible, and it will form a tight little ball. And this sealed compartment is the most energetically favorable state, which is what has been represented here. If you have a planar phospholipid bilayer, with edges exposed to water, then this is what happens. However, another possible structure is a micelle, which is something you might have come across when you would have studied some detail about how soaps or detergents, they work, right? Um, in that case, instead of a bilayer, you have um, a monolayer of lipids, basically, and the hydrophilic heads are facing the outside and the hydrophilic tails are at the center. And it is a completely sealed structure. Nothing can enter a micelle. Like you cannot really form an organism with a micelle. That's why there are two layers, and the phospholipid bilayer, which is planar in nature, will assume its most energetically favorable state when it is forming such a sealed compartment. And that is exactly why cells also form such compact structures, not just the outermost outermost layer, but instead the endomembrane structure as well, which is what is represented on the right. You have transport vesicles, you have the transgolgi, cis-median and transgolgi network, or any other sort of vesicle that might be formed during the cell's course of living. And all of these are made up of the lipid bilayers, and they usually try to adopt the sort of spherical structure to minimize the entropy and to form the most energetically favorable state. Uh, Here there is a certain concept. So I'm not sure if this was explained in much detail in the lecture, but basically, the idea that the membrane bilayer is fluid was understood by a technique called as trap, which is fluorescence recovery after photobleaching. So if you just look at the name, it means fluorescence is recovering after you have photo bleached it. Uh, we studied fluorescence, right? So previously, now what happens is during this experiment, if you have a certain lipid bilayer compartment and you photo bleach a certain, you expose one particular region of that particular sealed, let's say, cell to a very intense radiation. What happens is that the phospholipids that make up that region, they become photo bleached and they're no longer capable of any further fluorescence. And what you see is that when you photo bleach only one region initially, if you give it some time, you will observe that the phospholipids that were initially photo bleached are not just staying there. Some of them might be there, but a lot of them might also have, moved around in some other part of the cell, which means that the membrane bilayer is not rigid. There is movement among the lipids in that surface. You think of a surface and within, or if you just think of it as somewhat analogous. Say you think of a two-story building where the two floors are the two leaflets of the lipid bilayer. And within each of these floors, people that are, say there are 50 people in each floor, 50 people within the first floor will be able to move around anywhere within that floor. But the question that might come to your mind is that can people from first floor go to the second and vice versa? This is a slightly more advanced concept. It requires certain enzymes. There is something called as flippase, which flips lipids from one layer into another, but it's not the most common practice. It's possible, but it's not the most common practice. Normally, lipids move around in their own position, which means they can rotate. And apart from that, they can move within the same floor. And that gave the idea that there is some fluidity. The membrane bilayer seems to be sort of a fluid layer. So the membrane here that was there in the video is tagged with GFP so that we can see how exactly that fluidity is occurring inside the cell. Now, the composition of a membrane will be the major contributor towards dictating how fluid that particular membrane will be. Whenever there's a cholesterol in the membrane, it ends up stiffening the bilayer because cholesterol is a much larger structure. It does not allow for very easy movement, and therefore it's not it is a somewhat of a hindrance whenever it is present in the membrane. So depending upon what is the composition, the membrane properties vary, and in fact, it is the other way around as well. So you'll find a lot of questions like this when you look at questions for all of these competitive exams also, that if a particular organism is, say, you talk about the plasma membrane composition of a polar bear and compare that to some sort of some human being who lives in a very tropical area. The temperatures between the two are completely different, and depending on that, the not just the temperature, but the environmental conditions overall are quite different. Depending on that, there is a very high possibility that the composition of what makes up their plasma membrane would be entirely different. There would be a different percentage of saturated and unsaturated fatty acids, and that would be crucial to their survival as well. So you can read up a little bit on it. Not the most, you don't need to remember very in-depth details about it, but you can understand the concept that how is the difference coming about, what conditions are favoring, what type of membrane compositions. That would be a useful thing, I believe, for you to read up a little bit more on. So here, yeah, we talked about cholesterol making it more rigid, but apart from that, when we talk about phospholipids, there are majorly four types, particularly in the plasma membrane. So we have phosphatidylethanolamine, where the added, so the basic structure of it is the same. What changes is the attached group. So we have phosphatidylethanolamine, phosphatidylserine, phosphatidylcholine, and sphingomyelin, which are the four major types of phospholipids that are present. The different head groups are represented by different colors, as was shown here, and they are all derived from glycerol except sphingomyelin, which is derived from serine, sphingosine. So those are the different types of phospholipids. And here you see composition of mammalian lipids. There are glycerolipids, phospholipids, and sterols that are present in different percentages. You can look at it just for your reference, 65, 10, and 25%, versus mitochondrial glycerolipids, which are of a different origin. They are derived, they're believed to have been derived from bacteria. So they have fatty acids, glycerols, and phosphate groups. So you have PGCl, choline, phosphatidylserine, ethanolamine, etc. Their composition, their structure is a little bit different, and that is also believed to be the result of being derived from a prokaryotic origin. And then come membrane proteins, and we talked about it a little bit. Majorly, if you talk about, depending on the function of proteins, membrane proteins, they can majorly be of four different classes. They can be transporters, either allow only movement of the molecule into the cell or outside the cell. They can be anchor proteins, which hold certain components at certain positions, particularly relevant for the cytoskeleton. Then come receptors, which bind a ligand from outside and elicit a reaction downstream of it. And a different type of enzyme. So these are receptors, and these are also enzymes which are converting something inside the cell cytoplasm only. So proteins are composing approximately 50% of the membrane. And you see that there is an approximate approximately one protein is present for every 50 lipid molecules. So the ratio of abundance of the two is somewhat like this, 1:50. And membrane proteins can have many different functions depending upon what pathway or what process they are involved in. And here you see with the different types, we already talked about it. There are certain there are different classes along it. There are certain transmembrane protein, membrane associated, peripheral proteins. There can be lipid anchored or lipid linked proteins, or they can be attached to some other proteins as well. Peripheral membrane proteins, they associate with one side, but transmembrane proteins go across both of the layers of the lipid bilayer, both of the leaflets of the lipid bilayer. they traverse the two, and therefore they are said to be transmembrane. They are going across the membrane in comparison to just going through one lipid layer. So yeah, that was about the cell wall and the cell membrane, which are involved in the complexity and compartmentalization of cells. The next topic, and I guess the last topic for today before we get to questions, would be the cytoskeleton. So I think I introduced the cytoskeleton very briefly in the last session. If you just understand the terms, it simply means the cell's skeleton. But what has to be understood is that it's not just a simple sort of railway track that is present inside the cell. Not even the railway track. It's not just not equivalent to the building's metal structure that is just present there. It has very, very active functions within the cell. It is therefore believed to be, not just believed to be, but it is indeed dynamic in nature, and it has major roles to play in cell division, in cell communication, in sensing, a number of functions. Each of the different components of the cytoskeleton have very distinct functions that they perform depending on what process the cell is currently involved in. So when we talk about the cytoskeleton, immediately what comes to mind is the fact that it is offering some sort of structural or mechanical support. It is maintaining the cell shape. Certainly, it does so. It is required for the cytoskeleton to do so because just the cell membrane by itself, or the concept of the vacuole that you saw in plant cells, is not enough. It is maintaining the pressure, but the cell shape needs these proteins. But that is also not its only job. We'll come to it later. What we say is that the cytoskeleton is majorly made up of fibrous structures. They are not of the same type. There are three major classes, and they are individually involved in different sort of processes. The analogy that is majorly looked at is the fact that the fibers are believed to act sort of like a geodesic dome, and they stabilize as well as balance opposing forces. So they're forming the if I say in Hindi, it forms like a dhacha, the structure, the core structure of the cell. Apart from that, they support the concept that see, okay, if we just look at if we just explore the idea that when you think of a cell, when you draw a cell, let's just think of it that way, that when we were uh, everybody was learning about cells, the initial diagram that we all came about was that it's a structure with another little circle that is present in the center. So that is the cell with its nucleus. That was it. The more we understand, the more we get to know that the interior of the cell is extremely complicated, which is why it's a living, breathing entity by itself. So everything that you see inside the cell, every organelle that you see, every protein, every vesicle that you draw, that is not just floating about inside the cell. There are definite pathways, roads, or tracks, whatever you may think of. They are all crisscrossing, and they're forming this entire architecture or entire network inside the cell that is making sure that whatever is synthesized or whatever is made inside the cell is going to the place that it is supposed to go, and not just drifting about in infinite space. No, it is providing some anchorage, and this anchorage is not just restricted to organelles, but restricted to anything that is synthesized inside the cell. Let me just give you one example. There are some excellent videos. I'm not showing it right now, but sometime go and look up at how a neurotransmitter is moved inside a neuron. So if you've studied the idea of how a synapse works, you would know that basically there are two neurons that are forming a connection with each other, which is the synaptic junction. And whatever signal is in the previous neuron, the presynaptic neuron, is being taken over or is being communicated to the next one. Now, after the second neuron has taken up whatever information was given by the presynaptic neuron, the postsynaptic neuron is the second one. Whatever information has been given, or say in a neurotransmitter, let us think of dopamine. It is a particular neurotransmitter. After it has been taken up, does it just float about in the neuron? No. There are certain structures inside the cytoskeletal structures inside the neuron, which you can think of as these long sort of tracks or railway track-like structures. On top of that, there are certain motor proteins like Consider them equivalent to trains which are on a railway track. If the cytoskeleton is the railway track, the motor protein itself is a train or just a compartment of a train. And within that train, there is a suitcase which would be a vesicle, and that vesicle is carrying your neurotransmitter, which is dopamine, and it is moving along this particular railway track within the neuron, and it is being taken to the cell body of the neuron, which is where it is supposed to execute whatever its action is. So just an example, look up this video, you'll find it on YouTube. It's an excellent way to understand how the cytoskeleton works. That is how I was taught cytoskeleton as well, so I'm recommending the same. But yeah, basically they act as transport highways of the cell. They provide anchorage. They are providing, obviously providing mechanical support, and they are very crucial towards the normal functioning of the cell. And why they are so crucial, one of the reasons you'll understand it is because there's a very crucial role of microtubules, particularly in cell division. So um, if I just give you a brief idea regarding it, microtubules make sure that um, okay, since we have talked a little bit about chromosomes previously, if a particular cell has N chromosomes and they double during the process of cell division, which is why you have two N chromosomes within that particular cell, during the process where that cell needs to divide into two, it has to make sure that after everything inside the cell has been duplicated, each of the daughter cells is getting one copy of each. This making sure that we talk about how is the cell doing it. It has these mechanisms, these checkpoint or security mechanisms based. One of the ways in which it makes sure that each daughter cell has an equal copy of the genetic material is by microtubules. So microtubules attached to these chromosomes that have doubled and now they have separated after the anaphase, or during the process of anaphase. In fact, during metaphase, they attach this. I'm getting into a lot of detail, but just for your information, during the process of metaphase, they attach to the chromosomes and then they pull it apart and they make sure that there is one copy each that is being taken to the daughter cells. And there is something as crucial. There is a mechanism, not getting into detail of it, but there's a protein called MAT2 basically, which makes sure that each and every microtubule has attached to each and every chromosome that it is supposed to. Until that has been done, the cell will not proceed for cell division. And if obviously these are mechanisms that are in place under normal circumstances, which is why most people that you see around do not suffer from such severe congenital disorders. When these mechanisms are somehow bypassed and there are errors, and despite the unattachment of the microtubules, still the cell is proceeding for cell division, there is an unequal distribution of the chromosomes, or there is non-disjunction of the chromosomes. And that is why people suffer from aneuploidy, polyploidy, or other sort of conditions. So cytoskeletons, which you think of as just very just mechanical support for the cells, they have such details and such crucial functions by the cell as well. And there comes in their ability to be dynamic, because they will dismantle at one spot and they will reassemble at another spot to change the cell shape. So if you just think about the fact that within the cell, if you just think of the cell, and at the center there are all of the chromosomes which are aligned at the metaphase plate. When they have to undergo separation, the microtubule, which was initially quite long, has to shorten, right? It has to become short and has to pull apart. So there is a disassembly of microtubule that comes in. These are very, very detailed and excellent mechanisms. If it intrigues you, please go ahead and read it. You can refer to any cell biology book, particularly Karp and Cooper. These are very, very good books for cell biology, and they're very interesting with really beautiful schematic representations, if that's something that is appealing to you. Read up on those. And something as simple and as intricate as this is actually one of the reasons why certain drugs for cancer are designed in a way to stop microtubule disassembly, like taxols. Even though taxols are not the first line of therapy, they come much later. Because they have a lot of off-target effects, but taxols basically they interrupt this microtubule assembly, disassembly, or the dynamism of microtubules is basically interrupted. So those cells are no longer able to divide and you expect them to die. So this gives you an idea that how much research and how much studying something as simple as this can help you find cures to diseases which are so severe for the population around the world. So just I diverted a little bit, but just to highlight the importance of studying what the cytoskeleton basically is. So yeah, coming back to the slides. Here you are seeing that individual soluble units, which can differ based on the type of cytoskeletal unit you're talking about, whether it is microtubules, then they are tubulin. When it is actin filaments, there are F-actin, G-actin, globular actin, which is present, and there is intermediate filament as well. So these little protein molecules or these protein subunits, they assemble and they form these large filamentous polymers, and they perform the function inside the cell because individual tubulin or individual actin will not be able to do so. So you are seeing that here whenever there's a signal, such as suppose if you this is just a representation of very primitive cell that you think about sort of an amoeba, if you think about it, then when it has sensed that there is a nutrient source somewhere nearby, what it needs to do is it needs to form a sort of pseudopodia or false feed that we talked about, and it has to engulf that food material so that it can pick it up and use it for its deriving energy from the same. So for that purpose, it needs to reorganize its cytoskeletal subunits. So there has to be a disassembly of the filaments, and there has to be diffusion of the subunits, and they have to reassemble at a new site. So all of this is involved or is under the purview of the dynamism of cytoskeleton. So microtubules, they determine the positions of membrane enclosed organelles, and they are very, very crucial towards directing intracellular transport. Already something I discussed. Actin filaments, on the other hand, which are so commonly these are known as the why you might ask that why are the third group called as intermediate filaments? So they have alternative names for themselves as well. There are thick, thin, and intermediate filaments. So it's just based on the dimensions of those cytoskeletal elements. There's a lot of detail that you can go into. There's a way in which these are organized. So alpha, beta tubulins, they form dimers, and then these dimers are arranged one on top of each other. There's a certain number of tubes that are arranged at a certain angle to form a microtubule. It's a lot of detail. If you want, please go up and read about it on either Carp and Cooper that I already suggested. They are beautifully explained in these reference books. So microtubules are usually considered to be the reason behind membrane enclosed organelles and for intracellular transport. Then comes the actin filaments, which determine the shape of the cell surface. So they are majorly present right beneath the plasma membrane, which if you remember the previous diagram of the plasma membrane, there was a noodle-like structure which was present. So actin is usually one of the major contributors to this, probably the major contributor to it. They are localized right beneath the plasma membrane, and they are very, very crucial to whole cell locomotion. They have other functions as well. Okay, we'll avoid it for now, but there's actin and myosin, which are involved in muscle, basically in the movement of a muscle that you, whenever there's a contraction or relaxation of any muscle, how that happens is also something that involves actin very extensively. So there's a sliding filament theory. It's something you might have already studied at some point of time, but yeah, you can link these two and get some idea about it as well. And thirdly, intermediate filaments, they are majorly credited towards just providing providing mechanical strength and some resistance to any shearing stress which might come from the external environment. So yeah, now these are just comparative analysis of the three. While this has a little bit of detail, you don't need to remember it. I'll just go through it once for your reference. So microtubules, like I said, they have 13 columns of tubulin molecules. They form sort of hollow tube-like structures. The microfilaments or actin filaments, they have two intertwined strands of actin. So there's individual actin monomers, they form trimers, and they assemble one on top of the other in a sort of twisted arrangement. And intermediate filaments majorly are fibrous proteins that are formed into supercoiled into thicker cables. These dimensions of the three are also given. So you can see that they are 25 nanometers, which is why they're thick filaments. And they have of course, have a lumen. They are a hollow sort of structure. Microfilaments or actin filaments are approximately 7 nanometers, while intermediate filaments are somewhere between the two. Protein subunits for microtubules are tubulin, alpha and beta tubulin. Microfilaments are actin, and intermediate filaments majorly have keratin, which is one of the major cell types. Now, another link that I would want you to draw is not sure if any of you are very familiar with the common molecular biology techniques, but whenever techniques like qPCR or Western blotting, immunoblotting, these are utilized. Whenever you want to compare the expression of any XYZ, any protein between condition A and condition B. Let us assume you have condition A where you are just taking any particular cell population. Let's say we are taking, again, cancer cell population, and you are taking condition B where you're treating those cancer cells with a drug, any particular drug. Uh, and you want to see what is happening to your protein of interest. You perform these assays and you check the expression of your particular gene of interest using qPCR or using this Western blotting. Once you have checked the expression, what is always done in techniques like this is that you always need a reference. That suppose you find that expression in, let's take Western blotting only for ease of understanding, that you are observing that your gene of interest shows if you are expressing expression as X, then in case of the undrugged condition or the normal condition, you are getting an expression of 5x versus when you are adding that drug, you are getting an expression of 2x. So you are seeing that whatever treatment it was, it has reduced and it has become quite less compared to the normal untreated condition. So to make sure that whatever you have done is a result of the treatment and not because of any other changes, that maybe you took different amounts of cells or you made some manual error, etc. What is always needed is a reference or a, in other words, it's commonly known to be a housekeeping gene. Housekeeping genes are those which are considered to be, if you just understand by the name, you expect those to be so crucial to the cell's normal function that you always expect it to be present at nearly equal levels in all cells, or at least in the same cell population. So housekeeping genes mostly involve the use of these proteins. The reason I brought it up is because actin, particularly beta actin, is one of the most common genes which is used for normalization. In fact, beta tubulin, gamma tubulin, these are also very, very commonly used. I always use beta-actin as the housekeeping or the loading control. It is also known as loading control, which is used for comparison between the two. The reason I brought it up is to make you understand that their expression is believed to be constant and they are very crucial to normal cell function. If there is a very huge variation in their expression, maybe something else is affecting it, and therefore that needs to be studied in some other way. So yeah, coming back to the slide again. So microtubules, microfilaments, and intermediate filaments, we looked at the first three. The main function here is considered to be maintenance of cell shape, cell motility, chromosome movements in cell division, which I explained, and also organellar movements, which I also explained. Microfilaments have some similar functions, but there is also muscle contraction, cytoplasmic streaming, cell motility such as pseudopodia, and cell division. And the intermediate filaments majorly are the tension-bearing elements. They are anchoring the nucleus and other organelles, and they form the nuclear lamina. So what you see here in the immunofluorescence images are showing how these are arranged within the cell. So this will give you an idea that the interior of the cell is not, it's not like a soup. There is a network, a very intricate mesh. It is completely filled with microtubules. We just look here. Let's just zoom in at the microtubule image. You will see that it looks like the entire cell is made up of microtubules. It is so intensely populating the cell interior, and it has to because it is regulating such important functions. So this just gives you an idea of how relevant they are for normal functioning of the cell. Now, coming to redundancy. So that basically covers up the concept of cytoskeleton. Redundancy, okay, we'll just this. I think this was not taken up in the. Okay, we'll go over this. I think redundancy we can cover later. So yeah, the microtubules are made of protein subunits of tubulin. It is a heterotriangle made up of alpha has a site for binding to GTP. GTP bound to alpha tubulin is trapped at the dimer interface. Beta tubulin, the nucleotide on beta tubulin in contrast can be either in the GTP bound or GTP bound form, and it is exchangeable. And this is actually what there is something called as dynamic instability, which is a concept you can read up on in more detail. But basically what it does is, if you think of microtubule as a sort of pipe like this, there is microtubule disassembly at one end and there is microtubule assembly at the other end, which gives a sort of polarity to the microtubule. There's a plus end, there's a minus end. So on one end, there is always microtubule tubulin units which are dissociating. On the other end, they're associating and they're forming this sort of cycling. So there is a instability in the microtubule structure, which is what is making it dynamic in nature. So here you can see these are tubulin subunits and their, I believe, their experimentally determined structure, probably the crystal structure which was seen. And this is how an individual protofilament is formed. And these protofilaments are arranged around each other to form the microtubule hollow structure, as you can see. And this is the plus end and the minus end. And you can see that the beta tubulin is at the plus end, the alpha tubulin is the minus end. And like we said, the beta tubulin can either bind to GTP or GDP, and it is exchangeable, which brings about the microtubule dynamics. So like I just explained, the microtubule itself has structural polarity at one end, which is the minus end. It has alpha tubulins exposed, and on the other end, which is the plus end, beta tubulins are exposed. So that forms the structure. And here you have an example of microtubule organizing center, MTOC, which is centrosome is one of the MTOCs where microtubules are formed. And for migrating cells, their MTOC is located at the front of the cell because it is the direction in which microtubules are getting synthesized to direct the cell to move in that direction. This is somewhere else. And for actin filaments, on the other hand, like tubulin, each actin subunit has a binding site for a nucleotide. But in actin, instead of GTP, the nucleotide in question is ATP with ADP. So actin subunits assemble in a head-to-tail arrangement with, they also have structural polarity. They have two parallel protofilaments which are twisted around each other and they form a right-handed helix. Compared to microtubules, they are relatively more flexible, but they are cross-linked by a large number of accessory proteins. There's ARP23, there is alpha-actinin, beta-actinin, etc. There are a lot of proteins which are together maintaining the actins, the role of actin in maintenance of cell structure. So large-scale actin structures end up being much stronger than the individual actin filament. And here you can see what the actin filament looks like. It also has structural polarity. So there is a, when we say polarity, it means there is a difference between the two poles, right? For microtubules also, we saw there's a plus and there's a minus end. For actin filament also, we are seeing there is a similar plus minus end, which is observed. And this is again a sort of space filling model of what an actin filament probably looks like inside the cell in 3 dimensions. And again, this is just a representative image. In contrast to these, however, intermediate filaments, they do not have any structural polarity. Why is something you can explore from, again, those 2 books that I talked about, because of the way the individual filaments are assembled. They coil around each other, and the way they are effectively coiled around each other cancels the polarity. So the initial filament has polarity, but they arrange or they orient themselves opposite to each other, and that cancels out the polarity for intermediate filaments. And it's also not particularly required because they do not encourage that dynamism, which is seen for microtubules and for actin filaments as well. So that kind of covers up what was part of this week's lectures. Now coming to some of the questions. I'm assuming since nobody has interrupted me, there are questions from your end. If there are, please go ahead and ask. I will try to answer them. We talked about the plasma membrane. So the first question is asking about the fluidity of a cell membrane. They're being asked that fluidity would decrease with which of the following. So you want, so you need to understand that if fluidity needs to decrease, there has to be an increase in elements which are reducing the movements of the lipid, right? Movement of the lipids. So first is increase in glycolipid content in the membrane. Would it really affect the fluidity? Not really. Decrease in unsaturated fatty acids in the membrane. So if there are less unsaturated fatty acids, which means there are less fatty acids that are carrying double bonds and triple bonds, um, it means there are more saturated fatty acids, right? So that would not necessarily, if the enrichment is the same, you would not expect there to be much change if the unsaturated fatty acids are decreasing. Next comes decrease in saturated fatty acids in the membrane. That would also not really affect the cell membrane fluidity. But when there is an increase the unsaturated fatty acids in the membrane. So whatever was the initial lipid composition, from there, when there are more number of unsaturated fatty acids, which means there are more and more a double triple bond containing fatty acids which are being introduced, what it does is it limits the flexibility of the individual lipid molecules, and therefore by effect the fluidity of the cell membrane also gets severely affected. So option 4 is the correct option. And based on these lipid compositions, like I said, that depending on the environment in which a particular organism has to survive or has to adapt, the composition or the percentage of these lipids can also significantly vary. Um, Question 2 is about how plant cell wall is composed of A, peptidoglycans and cellulose, B, cellulose only, C, chitin and hemicellulose, or D, cellulose, hemicellulose, and pectin. So the correct answer is option 4. So if you think about the plant cell wall, it has cellulose fibrils, which are embedded in a sort of matrix like structure, which has hemicellulose and it has pectin as well. So that would be option 4. Um, then in question 3, there asked that which of the following statements is true? Gram-positive bacteria have only cell membrane. Not true. They have a cell wall and they have a cell wall that has a very thick peptidoglycan layer, which is why they stain blue when we are performing gram staining. Then second option is that gram-negative bacteria, they have an outer membrane surrounding the thin peptidoglycan layer. Yes, this is certainly correct. Then in option 3, we are asked that gram-positive bacteria have an outer membrane surrounding the thin peptidoglycan layer. No, obviously, because option 2 is correct, option 3 is not. And fourth option is that gram stain is retained by gram-negative bacteria. Also, this is not correct. So option 2 is the correct option that we are talking about here. Uh, in the fourth question, we are asked the secondary cell walls are present or in which of the following organisms. So we talked about secondary cell wall as well, how compression gives rise to it, and whether they're seen in animal cells, fungi, or viruses. So viruses are obviously not an option that needs to be at all studied here. Animal cells also mostly, under most circumstances, do not possess a cell wall. Neither do fungi. Fungi instead have a slime layer in a lot of cases. They have other adaptations to give them more rigidity. But here instead, plant cells in a lot of cases can have a secondary cell wall. So option A is correct. Then microtubules are made up of actin, myosin, alpha beta tubulin, or lipids. So actin obviously is making up actin filaments, or the myosin is not making up. Myosin has a different function. Like I said, in case of the sliding filament theory for muscle contraction, you would come across myosin as well. Myosin also makes up thick filaments, but those are different. They are not microtubules, so do not confuse the two. Alpha beta tubulin are what make up the microtubules, and we have studied that in a lot of detail as well. So option 3 is correct. Then in question 6, we were asked that what is the function of vacuoles present in plant cells? So we kind of encountered a similar question in the previous week's assignment as well, but here obviously you can see the options are different. The first is that it supports and protects the cell. It controls what enters and what leaves the cell. It controls the cell volume, or it stores water and mineral ions. So it can somewhat provide the turgor pressure and the rigidity of the plant cell that we talked about, but one of its major jobs is also to store water inside the cell, which is what is effectively maintaining the turgor pressure of the plant cell. But inside the vacuole, what you have is a storage of water and mineral ions, which is useful to the plant cell for its normal survival as well. So option D would be the correct option there. Then in question 7, okay, so this actually brings the concept of redundancy. I think I missed it, but yeah. So I think we had highlighted redundancy the previous week as well, that redundancy is not something that is supported by a biological system under normal circumstances, because it would end up in very unnecessary and wasteful investment of energy, which is not something a cell would ever try to do. It will always try to conserve energy and direct it in a way which is the most economically, when I say economically, I mean energetically most desirable, most useful. So when we see that here, the example citing is somewhat the same, that if you consider the Indian cricket team and you have MS Dhoni, Rishabh Pant, and Sanju Samson all playing in one team, what is it an example of? It means that when there are three different things which are performing the same function, it happens in cells, but it is not the desirable thing. It is regulated in such a way that it can be controlled. And you don't, like, for example, if you think about the cytoskeleton, it might appear that the mitotubules, the actin and intermediate filaments, they are all part of the cytoskeleton. But are they all redundant? No. They're all performing their own functions, and each of them is necessary for the cell to survive. So it's a different scenario. But the answer to this question, like you said, that if they were all performing the same function, that if they were all doing the same thing, they were all mechanical support for the cell, it would have been something that is redundant. So they exhibit the phenomenon of redundancy. Then comes dynamic instability, which is something I have mentioned today in question 8. The dynamic instability is an inherent property of A, microtubules, B actin, both A and B, or none of the above. So technically, while you can see some sort of dynamic instability in actin as well, you will see disassembly and assembly in case of actin as well. But actin has a large number of protein complexes that are associated with it too. So when we talk about dynamic instability, it is believed to be an inherent property of microtubules. So there is constant assembly and disassembly at the microtubule ends, the plus and minus ends, and therefore dynamic instability is usually associated with them. I don't have a more clear answer for it at this point, but when inherent property is associated with dynamic instability, it is credited to microtubules only. Then in question 9, we are asked that which of the following cells they lack a cytoskeleton? Prokaryotes, eukaryotes, both A and B, and none of the above. So this is something that is a little debatable because if you look at the lectures, it says that prokaryotes, they lack a cytoskeleton. But in the sample problem, this was the given answer that it is none of the above. So in my opinion, it could be prokaryotes, but there are certain proteins which are believed to be analogous, somewhat similar to that of the ones which we see in eukaryotes. I think it was mentioned in the lecture as well, like FGSZ. So proteins like that, there are similar analogous proteins. You do not exactly have a microtubules, or you do not have actin exactly like that, but there are other sort of structures which are somewhat involved in maintenance of cell structure, in locomotion a little bit. So somewhat similar proteins are there. You can look them up for your reference. So when we look at certain examples like these, then then you can say that prokaryotes do not, while they do not have a very well-defined cytoskeleton, they also do not lack it. So under such circumstances, the more acceptable answer would be option D, that neither of the or none of the above exactly lack a cytoskeleton, but instead there are some homologous proteins which can be found in prokaryotes as well. In option D, question 10, we are asked that which of the following is a function of the cytoskeleton? So is it maintenance of the shape of the cell, positioning of the organelles of the cell? Yes and yes, both of them, which is why option C is correct, that both A and B are the function of the cytoskeleton. And apart from this, like we have already explored, there are other diverse functions for the cell as well. So I think, yeah, we have already covered gram staining. So I guess that covers everything I had to bring in for today. So in case anybody has any doubts, please go ahead and bring them up now. I would be happy to answer them. And if not, then we will wrap up today's session. So please go ahead and let me know. If not, you can reserve your questions for the other sessions as well, or you can put them in the discussion forum. And then I guess we are ending the session for today. Thank you so much for joining, and I will see you again in the next week. Thank you, and good evening to everybody.